Hi, welcome to yet another session of biology classes with Lakshmi Chandra. As you know, we were dealing with different genetic disorders. We have completed a few genetic disorders. So today, let's focus our attention on yet another genetic disorder named thalassemia. The name of the disorder is thalassemia. From the name, can you just guess what this disorder is or in which part of the body this disorder is actually affecting? From the word thalassemia. Do you find that there is a relation when you say the word thalassemia and anemia? Right. So it's something related to anemia. And when I say that thalassemia is related to anemia, I hope another genetic disorder's name is also coming into your mind. Yes, that is sickle cell anemia, which we completed in the previous session. So if you haven't gone through that, you can log on to the channel and get the details of the genetic disorder sickle cell anemia. And after completing this topic, we'll just compare the disease sickle cell anemia with thalassemia. At that point of time, if you find doubtful, again, you can go back and refer, you can subscribe the channel and go back and refer to this disease sickle cell anemia so that you get clarity. Let's focus our attention to thalassemia today. This thalassemia is a genetic disorder which is autosome linked. So it is autosome linked genetic disorder. Explanation of this autosome linked disorders, what they are, why they are called so, etc. were dealt with in the previous episodes. But still, to have a refresh, I'll just tell you, there are diseases which affects our sex chromosomes and there are diseases which affects the rest of the chromosomes other than sex chromosomes. So those diseases which affects the rest of the chromosomes or autosomes, you call that as autosome linked genetic disorder, and one example for that is thalassemia. So when I say it is the autosome, we should know which chromosome. So thalassemia is as a result of mutation happening with chromosome number 11 and chromosome 16. So when there is a mutation with chromosome 16 and chromosome 11, that disease is called as thalassemia, and all diseases related to chromosome 16 and chromosome 11 will not result in thalassemia. But to make it clear, if in chromosome 16 the genes HBA1 and HBA2 are affected, then it results in thalassemia. Again, if you take chromosome number 11, there will be plenty of genes, but if among those genes, if HBB is affected, then again it will result in thalassemia. What is this HBA1, HBA2 and HBB? To put it in very simple words, these are genes. Where are they located? On chromosomes. On which chromosomes? These two, HBA1 and A2 on 16, chromosome number 16, and HBB on chromosome number 11. Now let's go into detail about what is this HBA and HBB. In order to understand that, we should know again what is anemia. I have already explained in the previous session, but still, so anemia is a disease which affects our hemoglobin. You know, regarding hemoglobin molecule. So this hemoglobin molecule is having a protein part called as globin. So let me just write here. We have in our red blood cell, we have hemoglobin, and this hemoglobin is having a protein part, and that protein part is called as globin. What is this heme? This is the iron part. Okay, so our focus is on this part, globin region of hemoglobin molecule. So this globin is actually a protein. As you know, every protein is synthesized from a gene. This globin protein is also synthesized from a gene. But in this hemoglobin molecule, it is made up of two types of globin proteins. Hemoglobin molecule is having a globin protein, and that globin protein is of two types. What are those two types? You say it as alpha globin and beta globin. So hemoglobin molecule is made up of a globin protein, and that globin protein is having two different forms of protein, that is alpha and beta. Explanation regarding the structure of hemoglobin and what this alpha and beta chains are were also dealt with in the sickle cell anemia session. So it will be better if you log on to that first. Now here, so globin is having two chains, alpha globin and beta globin. They together combine with iron and they form hemoglobin. So I hope now what hemoglobin is clear to you. Now come back to our session here. See, alpha globin. This alpha globin, this is a protein or a polypeptide, and this protein is synthesized from a gene. From which gene it is synthesized? It is synthesized from a gene called as hemoglobin A. So hemoglobin A, HB is for hemoglobin and A here denotes alpha. So how can you denote the gene for synthesis of beta globin? The gene will be HB for hemoglobin and B for beta chain. So HBA and HBB are the genes responsible for the production of the proteins alpha globin and beta globin respectively. I hope that point is clear to you. These genes HBB, hemoglobin beta, is present in chromosome number 11, and the genes HB alpha 1 or A1 and HB A2 are in chromosome number 16. So from here you can understand that there is only one gene responsible for the production of beta globin, but there are two genes responsible for the production of alpha globin. So for alpha globin you have two genes. They are named as HBA1 and HBA2. These two, they are the genes, and these two genes are responsible for the production of alpha globin protein. Whereas HBB gene, it is responsible for the production of beta globin protein. And alpha and beta together, they are there present in hemoglobin. Actually, there are two alpha proteins and two beta polypeptides together forms that globin part of hemoglobin. So this much is clear to you. So these are the names of the genes and where they are present is shown here. Now come to the disease thalassemia. When there is a defect happening with respect to these genes, it results in thalassemia. So what kind of defect is that? If this gene is mutated or that gene is deleted, if say HBA1 is deleted, what will happen? This alpha protein will not be produced. It will not be produced at all. If HBB gene is not there in chromosome number 11, if that gene is removed, what will happen? The person's hemoglobin will not have the beta chain. So again, abnormal hemoglobin will be produced. So hemoglobin, if it is abnormal, it will result in anemia. So here we have learned anemia results in deficiency of iron, Anemia can also result due to the deficiency of globin. Not deficiency, but abnormal production of globin part. So anemia can be because of many reasons. Accordingly, its name differs. In simple terms, if we say anemia, it is because of the deficiency of iron. If it is thalassemia, it is because there is deficiency of alpha or reduced production of alpha globin or beta globin protein. And why there is reduced production? Because there is abnormality in the gene for its production. Now, since this is a genetic disorder, it is passed on from parents to progeny. Now, what is the chance of the progeny getting the disease? See here, the progeny or the young ones will get the disease only if both the parents are carriers of the disease. This term carriers were also explained in the previous sessions. What is the meaning of carriers, affected individuals and all, where again it was already explained to you in the previous sessions. If you can log on to this channel of biology classes with Lakshmi Chandra and go to the playlist and play one after the other, things will be very clear to you. Still, if you have doubts, please feel free to ask through the comments link which is there in the YouTube. So come on, let's move on. So this is a recessive disorder. Why this is a recessive disorder? Because the progeny will get the disease only if both the parents are carriers. So we can say thalassemia is an autosome linked recessive disorder. So these two features of thalassemia is explained now. Why it is autosome linked? Because it is related to chromosome other than sex chromosomes. So which are those other chromosomes? Is chromosome number 16 and 11. The genes are HPA1, A2 and HPB genes. Now what is the reason for the disease? Mutation or deletion of this gene. So what it will result in? It will result in abnormal haemoglobin. Okay. Now the question is, what is the difference between thalassemia and sickle cell anemia? Thalassemia, why it is caused? Due to mutation or deletion of this particular gene. The gene itself is absent there. So that means the number of alpha protein produced or beta protein produced is less because the gene itself is not there. So we can say thalassemia is quantitative in nature. It is a quantitative defect. It is a quantitative disorder because the whole thing is not present. The protein is not present at all. But in sickle cell anemia, if you have seen the previous sessions, it will be clear to you. In sickle cell anemia, alpha, beta proteins are all present there. But then, in the beta protein, in the beta globin, one particular amino acid is replaced by another. This glutamic acid is replaced by valine. So there is no change in the quantity. But what happened is change with the quality. One is replaced with another one. So the quality is changed. So our RBC became sickle shaped and it resulted in anemia called as sickle cell anemia. That's it. So this is a qualitative change that is happening with the hemoglobin, whereas thalassemia is a quantitative change happening with hemoglobin. Now can you guess what is the other difference between thalassemia and sickle cell anemia? Thalassemia can be due to the deletion of any of these genes, isn't it? So both alpha globin and beta globin are getting affected in the case of thalassemia. But what about our sickle cell anemia? There the defect is only with respect to the beta globin chain of hemoglobin. Only with beta globin chain. Nothing to worry about alpha. So here sickle cell anemia, only beta chain of hemoglobin is involved, whereas in thalassemia, alpha chain and beta chain are involved. So this is the reason. Now, let me explain to you a little bit more here. I told you that alpha and beta chains can be affected in the case of thalassemia. Okay. We have learned that every gene has got two forms. What do you call that two forms as? Very good. Alleles. Every gene has got two forms. Those two forms are called as alleles. One form comes from our father, another form we get from our mother. Okay. In this particular case also, you have the gene, say HBA. HBA1. This is a gene. HBA1 is a gene. It will also have two forms. HBA1. I'm putting here a slash, and another one HBA1 without any slash I am putting. So these are the two forms of this particular gene. Okay. Again, the other gene, that is HBB. HBB. This is another gene. This can also have two forms. Again, I am representing it as HBB slash and simply HBB. Two forms I am writing here. Okay. This is simple notation. This simply I have wrote here. So one gene, two forms. This is there. Again, two genes are there. I have written like that. Similarly for HBA2 also it will be there. So I am writing here HBA2. This gene will also have two forms. HBA2 slash and HBA2. So here, see how many alleles are there? 1, 2, 3, 4, 5, 6 alleles are there. Among the 6 alleles, how many alleles are there for alpha globin? For alpha means A, that is 1, 2, 3 and 4. There are 4 different alleles present for alpha globin chain, but there are only 2 alleles. There are only 2 alleles present for beta globin chain. That means in a person's body, you will have how many alleles for hemoglobin? You will have a total of 6 alleles for hemoglobin. Only if all the 6 alleles are having the defect or mutation, the person will get the disease thalassemia. That is why it is written as a recessive disorder. Whatever alleles the person is getting, the offspring is getting, is defective from his parents. In that case only the person will get thalassemia. If a person is having among the 6 particular alleles defect in the gene HBA1. HBA1 is defective, means these 2 alleles are defective. Other alleles, it is okay. There is no problem with the other alleles. The person will have mild thalassemia only, or the person will not show any symptoms of thalassemia at all. But the person is having the disease because this gene is defective. In that case, you call the person as a carrier. Okay. So in that case, the person is a carrier. If these genes are also affected along with HBA1, if HBA2 is also affected, then there will be mild to severe case of thalassemia. If all the 3 genes are affected, what happens? The person is getting the real disease, thalassemia. Survival of such kind of persons will be, chances are very less. stillbirth and all results in this particular case. Stillbirth, baby will be born but cannot move and all. Stillbirth it is called. Dead baby will be born. God. Like that. Okay. So it depends upon how many genes are getting defected. The severity of the disease increases likewise. So it is totally a quantitative approach. If one gene is affected, others are okay, the person can survive. The person, it becomes very severe only if all the genes are defective. So I hope you understood regarding the disease thalassemia, and it's very important for you to keep it in mind that this is totally a quantitative disorder, whereas our sickle cell anemia was qualitative, and in sickle cell anemia only the beta globin chain is affected, whereas in thalassemia both alpha and beta chains are affected because of the defect or mutation in their respective genes. If you have any doubts, please feel free to clarify it with me. Thank you for now.